or for any technical issues, and the Q&A box to post your questions. Due to large number of participants, your camera and microphone are disabled. If you'd like to speak upon invitation by the chair, please raise your hands and we'll enable you your microphone so that you can speak. So with this housekeeping issue, let me introduce myself first. I'm Iyasu, working at Addis Ababa University, College of Health Sciences, Center for Innovative Drug Development and Drug Trials for Africa. I would like you all to welcome to this webinar organized by the Clean Ops Training Program, CDT-Africa, Addis Ababa University. Let me share my slide. Hope you can see my slide. Well, let me introduce you first what Addis Ababa University is. Addis Ababa University is the largest and oldest university in Ethiopia. Currently, it has enrolled over 53,000 students. The university has got over 73 undergraduate and 293 postgraduate programs. Addis Ababa University ranks 6th in Africa, and it's among 400, 500 universities in the world. Addis Ababa University has got the largest referral hospital in the country, and it hosts 3 World Bank supported African Center of Excellences, one of which is CDT-Africa. CDT-Africa is one of the 3 World Bank supported African Centers of Excellence hosted by Addis Ababa University. It's located within the premise of College of Health Sciences, or Ambassa Specialized Hospital. The objective of CDT-Africa is to support equitable access to drugs, diagnostics, vaccines, as well as other interventions like complex interventions, and bring about sustainable development in Africa through high quality capacity development for innovative drugs and therapeutics.
I'm sorry for the interruption. So
yes,
we are not looking at your slides. So should I share it from this end?
Oh, they're not okay. I thought I have shared already.
Yeah, but they are not visible. Yeah,
it's not visible. Oh.
So shall I share it from this end?
If not possible from my side, probably yes, you could do that.
But you have to stop sharing. Yeah.
Okay. What's wrong with me? Okay. Let me stop sharing. How about now? Is it coming?
It's saying Prof. Iyasu has started screen sharing, but
not yet
your
coming.
screen is not yet visible.
It's not visible. Okay. For now, it probably you could share.
Yeah, so I've shared.
Okay. Thank you. Thank you. So could you cut this slide slide 2, and then slide 3. Thank you. So as CDT-Africa focuses on medical discovery, it's working mainly on development of drugs, vaccines, and diagnostics. Could you share slide 4? Okay. Sorry, slide 3, just back to slide 3. Yeah. So its core functions include both research as well as capacity building activities. Slide 4. I think I'm missing one more. The previous slide. Yes. So CDT-Africa, to realize its ambition, it has launched MSc program in clinical trials and PhD program in translational medicine. So far, the MSc program has enrolled 4 batches of students with a total number of 77 students from 9 African countries, including Eritrea, Ethiopia, Zambia, Kenya, Uganda, Tanzania, Malawi, Rwanda, and Liberia, of whom 43 have graduated today. The remaining 34 students are either doing their MSc thesis work or the required courses. The PhD program in translational medicine has got 3 tracks: drug development, diagnostic development, and vaccine development tracks, and has so far enrolled 1 batch of students with a total number of 9 at the moment. CDT-Africa is a pioneer to start postdoc programs in Addis Ababa University in particular, and in Ethiopia in general. On top of these advanced level programs, CDT-Africa is involved in offering several short courses which help develop capacities in clinical trial and translational medicine. Next slide, please. In summary, CDT-Africa has trained over 700 students to date. It's involved in product development work. Over 300 scientific papers have been published by its core faculty since its establishment. It has developed new educational programs. It has established its own research infrastructure and is involved in several research and training projects funded by different funding organizations like EDCTP, NIHR of the UK, and others. It has also created job opportunities for over 100 Ethiopians. Next slide, please. Clean Ops training program is one of the short courses developed and conducted by CDT-Africa. Clean Ops focuses on developing and or strengthening capacity to conduct clinical trials efficiently. Clean Ops training program is a competency-based online training program designed mainly for study coordinators in Africa. This program has been given for the 4th iteration by CDT-Africa. It has so far trained close to 300 study coordinators from more than 27 countries. But this program is a 10-week course, 8 to 10 hours per week. Next slide. Next slide. CleanOps training program syllabus has been anchored in the TDR, TDHN competency framework. PDPs, CROs, fellows, FCDs, and others have input in this syllabus. It focuses on practical recurring problems in African sites, and of course covers all relevant domains of clinical trial, which will enable to conduct trial easily. It's an intensive on-job training for study coordinators. It follows innovative training approach, M&E efforts to ensure its practicability. Next slide. CleanOps covers various domains of clinical trial in depth. It has got 10 lessons. The first lesson introduces clinical research operations. Lesson 2 deals with data management and biostatistics. Lesson 3 focuses on study design and protocol development. Lesson 4 deals with project and financial management. Lessons 5 and 6 focuses on conducting a trial as part 1 and 2. Lesson 7 deals with closing out and reporting a trial. Lesson 8 deals with working with external partners. Lesson 9 focuses on quality systems, audits, and inspection. And lesson 10 deals with pharmacovigilance. Next slide, please. CleanOps training program involves innovative approach using Moodle platform with voice-led presentations and different tasks given to trainees. Next, please. Live tutorial is given on Thursdays for 1 hour, and it provides the recommended as well as in-depth resources to the trainees. Next slide. Assignment is given to develop risk management plan for the trainees as a group work in which they identify the risk, analyze the identified risk, come up with mitigation plan, and develop risk management documents. Next slide, please. This slide shows for further communication. So you can make use of these websites in order to know more about this training program as well as CDT-Africa at large. Next slide. Okay, thanks a lot then. Now, let me come back to introduce to the panelists for our webinar. But before we go to the introduction section, conducting clinical trials efficiently, as you know, looks very difficult in the African setting. There could be a lot of reasons for these problems. Delayed ethical approval process could be one. Delayed clinical trial authorization process as well could be another. Slow participant recruitment rates. Inadequate facilities available for conducting the clinical trials. Inefficient trial facility procurement process in African setting. Lack or inadequate capacity in conducting clinical trials, and many more challenges we have. How do we solve this and other problems associated with inefficient clinical trials in the African settings? Professor Thomas Jackie and Dr. Pavel Mzgonov are willing to share their experience on moving towards efficient clinical trials. May I use this opportunity to thank both panelists for agreeing to share their rich experience with our audience. Let me introduce Prof. Jackie and Dr. Mzgonov. Dr. Jackie, I wish I could share one slide for each, if it's possible. Shall I try?
Yes, go ahead, Prof, please.
Okay. Does it move?
We don't see it yet, but it says you are sharing, you have started sharing.
Oh, unfortunately, that means I'm not able to. I don't believe that you have this slide. So let me introduce them verbally then, if that's not possible. Sorry. We have two panelists for today's webinar. Let me first introduce Professor Thomas Jackie. Professor Jackie is Professor of Statistics and Program Leader at the MRC Biostatistics Unit at the University of Cambridge. He is a co-investigator of the MRC's Trials Methodology Research Partnership and co-leads its Adaptive Designs Working Group. His work focuses on efficient trials using adaptive and Bayesian methods. Then we have the second panelist, Dr. Pavel Mzgonov. Dr. Mzgonov is an advanced NIHR fellow in medical statistics, working at the MRC Biostatistics Unit at same university, University of Cambridge. He develops more efficient adaptive clinical trial designs and supports their implementation in practice. His statistical interests range from phase 1 dose escalations trial to the later phase master trials and quantitative benefit risk assessment. He leads the statistical designs of several publicly and privately funded trials. We shall be honored to have such distinguished panelists for this webinar. Let me call upon first then to share his experience, Professor Jackie. Professor Jackie, the floor is yours. Thank you so much indeed.
Thank you very much for the invitation and the kind introduction. I'll share some slides. If you can't see them, let me know. You should be able to see them now. David, can you?
Yes. Well, I can see them.
Excellent. Great. So what we're going to do is we're going to talk a little bit about why we need more efficient clinical trials, introduce what we mean by adaptive designs, give you a couple of commonly used adaptive designs, and then talk in a bit more detail about different examples where adaptive designs have been used and the experiences we've had with them. So just to start off, why do we even need adaptive designs in the first place? It's basically because at the moment, attrition rates for new developments are pretty poor. When we're looking at phase 2 clinical trials, over 80% of trials conclude that the treatment is not good enough to investigate further. So either because it's not efficacious or because there is some safety issues with the treatment. You could argue that in phase 2, that's not too bad because it's the first time that we look at efficacy. However, when we look at phase 3 and regulatory submissions, we still find that about 50% of trials do not end with the conclusion of, yes, this treatment should be used to the broader public. And that, for me, is really where the problem lies, because phase 3 trials are often called confirmatory clinical trials. So they are supposed to confirm what we already know. And clearly, half of the time, we don't know as well as we thought we did. When we look at the reasons why these different, why the trials fail, you can see that in phase 2, the majority is efficacy, followed by safety and other reasons. About a third of the trials stop because of safety, about 2/3 because of efficacy. In phase 3, we have more certainty about the efficacy. So now only about half of the trials conclude that the treatment isn't efficacious, but there's a rather large safety concern or an increased risk of safety issues in those confirmatory studies. When we look in more detail why these trials fail, it's often because we're taking forward treatments that we shouldn't have taken forward in the first place. We're looking at the wrong patient population. Maybe the treatment is working well in part of the population, maybe based on some biomarker or maybe gender. But because we're investigating in the whole population, we get a diluted treatment effect and are unable to show that the treatment is truly efficacious. And finally, it's about poor precision, about safety, about dosing of the treatment. And so the question then arises, okay, what can we do better? Well, first of all, if we look at the poor precision cause, this is a question that should have been answered in early phase studies, in phase 1 and phase 2 studies. And hence, rather than trying to be very fast and very clever and jumping straight into the expensive phase 3 confirmatory studies, maybe should be a bit more careful during the early stages of the development to avoid having poor precision on those crucial parameters. And the other option or the other thing to consider is to think about adaptive and Bayesian designs. So that raises the question, what do we actually mean by an adaptive design? Well, the idea is actually very simple. The idea is, in an adaptive design, we modify an ongoing clinical trial based on reviewing data that have been collected within the study and make a decision at the interim analysis in order to enhance the flexibility of the study. Now, this is not just done randomly, this is done by design. And so we're thinking before we even start the study that we might want to make a particular change to the study as it goes on the basis of the data that we accrue. So that change might be, we might think about changing the sample size, we might think about stopping a treatment either because it's working great or it doesn't work so well, or we might choose to add or drop different treatment arms in a study that has multiple different arms. All that sounds very straightforward and simple, and the tricky aspect is we want to do that without undermining the study's integrity and validity. So how does a typical adaptive design look like? Basically, you spend a significant amount of time planning the study and fixing the design. Then you start your study, start recruiting patient, recruit, observe, until you have a certain number of patients observed, at which point you look at the data of the patients so far and make a decision at an interim analysis. And at that interim analysis, we might think about stopping the study, again, either because it's hopeless, the treatment just doesn't perform well enough, or it's doing so great that we already can conclude that the treatment is working brilliantly. We might want to change the sample size because our assumptions initially have been incorrect. We might add or drop arms and many other adaptive features that we could consider. And we continue on with this process until a final analysis, unless we have stopped beforehand. So what I'm going to do next is, based on the paper that's displayed at the bottom here, I'm just going to talk briefly through a couple of different adaptive designs and the kind of question that they are designed to answer. And then Pavel will follow up with a couple of examples. So for example, if my question is, what dose is safe, or what is a safe dose? A good method to use is the Continual Reassessment method, or CRM for short. The CRM design is a phase 1 dose finding design that uses an iterative process incorporating all trial data that you have accumulated so far to decide which dose the next group of patients, the next cohort of patients should receive. And you continue on until you reach the highest dose that is considered acceptably safe. The advantage of using a CRM design is that all trial data is included in the decision making. We're predefining what we consider being an acceptable risk of toxicity. So we predefine what we are looking for, and it results in a high probability of finding a good dose, a dose nearest to the maximum tolerated dose. The drawback, as so often things come at a cost, there is additional complexity in designing the trial. So rather than using the commonly used 3+3 design, which requires no thinking whatsoever, we're going to have to think about some of the specifics of the design before we can get started. Therefore, it will require input from a suitable trained statistician, which as a consequence sometimes means that clinical investigators might see the process as a black box. If our question is to decide which treatment is the best treatment, so if I have multiple different treatments that I might consider, and I want to find out which treatment is best, I could consider either multi-arm, multi-stage design, or MAMS, or a response adaptive randomization called RA. In a MAMS design, we simultaneously compare multiple experimental treatments with a common controlled group and allow the trial to stop early, either for futility or for efficacy. In a response adaptive randomization, in a design using response adaptive randomization, we change the probability of patients receiving a particular treatment on the basis of the accumulating data. So imagine we have 3 different experimental treatments, and it turns out that after the first 10 patients, that the first treatment seems to be doing much better than the other 2. Then for subsequent patients, we'll give them a higher chance of receiving that first treatment in order to ensure that many patients get the treatment that looks to be most promising. Potential benefits and drawbacks for these designs. If I use multiple treatment arms and compare them to a common control, I've reduced my total number of patients required from running separate trials because I only need one control group, and that results in a massive decrease in the total sample size required. In addition, because I'm allowing early stopping, I expect the sample size to be even lower than this. There's also an ethical angle to it, because obviously if I stop treatments that aren't looking very good, I'm increasing the chance of future patients to get better treatment for both the multi-arm, multi-stage design, as well as for the response adaptive randomization. As before, this is more complicated than your traditional two-arm randomized controlled trial, and so there's additional complexity, additional time required, and it will require suitable trained statistical input in order to get things right. In addition, because we can stop trials earlier, there is no certainty about the maximum about the total number of patients that we're going to have to recruit. Imagine you plan a study with 400 patients, but halfway through, you have already enough information to conclude that the treatment is working. You end up with only 200 patients in your study. And so that's where this uncertainty comes in. My argument would be, however, that that's a good thing. It just means that we need fewer patients in our study. The other drawback, or the other thing to consider, is that because we require data in order to make modifications to our trial, our endpoint, our primary endpoint, needs to be relatively quickly observed, relatively quickly compared to the recruitment rate. If I'm interested in answering the question, does my treatment work in the first place? A good option would be a group sequential trial, which is basically a two-arm randomized controlled trial that allows at an interim analysis to stop, either because the treatment works or because it's hopeless at that point. Quite often, this is then paired with sample size reassessment, which effectively means I'm going to use the data in my study to update my nuisance parameters that I have used in my sample size calculation. So imagine I'm looking at comparing two normally distributed, trying to compare two groups using a normally distributed outcome. Then I'm going to have to make an assumption about the variability of that outcome. And at the onset, before I start the study, I might have a lot of uncertainty about what that value is, while in the middle of the study or during the study, I have actual data in the relevant patient population that I can use to estimate what that nuisance parameter, that variability in my outcome should be, and hence I can update my sample size calculation on the basis of these improved information. What are the potential benefits? Well, group sequential designs reduce the expected sample size over a traditional two-arm trial. You can do blinded and unblinded reassessment. So depending on whether you're going to look at unblind your data anyway or not, you have the option to do sample size reassessment. And I would argue that this is probably the most commonly used adaptive design using sample size reassessment. And I would also argue that for any study, you should at least consider whether sample size reassessment is sensible. And the big advantage of doing so is that you will end up with the right number of patients at the end of the study, rather than potentially ending with an underpowered study. As before, there is prices to pay, additional complexity you need to think about, the specifics of the design, you need some trained input. For group sequential design, there is a cost for stopping early, and that is the maximum sample size. So in the situation where you do not stop early, you might have to increase the sample size. So there's an increase in the maximum sample size. That increase is about 10% in the total sample size. So if you compare a two-arm randomized controlled trial to a group sequential design with two arms, you would need a maximum increase of 10% in the sample size. The benefit is that you would expect to decrease your sample size with the group sequential design by about 30 to 40%. And similarly to before, because of that potential to stop early, we have uncertainty about when the trial will end. With that, I'll pass you on to my colleague Pavel, who's going to talk to you through 3 adaptive design examples. Pavel, over to you.
Thank you. Just 1 second. So again, hopefully you can see my screen now, and thank you very much for the opportunity to contribute and for your kind invitation. So exactly as Thomas just said, what I'm going to do, I'm going to go a couple of examples that we have been involved with over the past years on how adaptive trial designs were implemented in practice. And we're going to cover different areas of the questions that Thomas covered in his introduction. So our first example today would be from phase 1 dose escalation trial called Forwards trial that takes place here in the UK. And this trial is about defining the safety of an agent. So this trial is answering the question, what is safe? So the setting of the trial is in the addicted patients. So addiction is one of the major challenges UK is currently facing. And one of the ways to treat the opioid addiction is with substitute therapy. However, so instead of getting the actual illegal substances, the addicts got some medical substance under the supervision of a medical professional. So it goes on for quite a bit, part of their lives, or maybe for their whole life. So, and it's quite hard for these patients to withdraw, and only few of them successfully complete detoxification and don't need this substitution therapy at all. So one of the ways to reduce the burden of withdrawal for these patients, and this is the hypothesis that clinicians wanted to test in this trial, is to give this drug called baclofen that is likely to improve withdrawal symptoms. And this evidence comes from other addictions, specifically from alcoholism addiction. However, the baclofen, this drug, was never tested in combination with methadone, and methadone is exactly the substitution therapy that currently opioid addicts take in the UK. So the objective of the trial was to establish safety, whether it's safe to give baclofen with methadone, and what doses of baclofen are safe to give with what doses of methadone, because different patients got different doses of methadone, which also imposes a challenge. So our solution to answer this question, what are the safe doses, was to propose a model-based dose-finding design, similar to the continual reassessment method, CRM, that Thomas mentioned earlier today. So the fundamental idea of a model-based dose finding is to assume a working model for the dose toxicity relationship. So in the left-hand side here, you have the risk of toxicity for particular dose J. And in the right-hand side, you have a function of a dose and some parameters that we're trying to estimate with our data. So what we're doing here is we link all doses together with some functional form. And we do that because we assume that the more drug we give, the more toxic it is. And we would like to incorporate these assumptions, this knowledge, into our decision making. So we know that the risk of toxicity will increase with the dose, and that information about the current dose can also give us information about how risky neighboring doses are, doses that are less toxic and are most toxic. Because we link those together, this model-based approach results in a more efficient use of data, because we benefit from the all information collected in the trial. It's also quite flexible and can be liked for various reasons. One of them that is that it can deal with changing cohort size that will not be always well predicted at each point. And one of the reasons that we like the model-based design for this particular opioid detoxification trial is it can be extended to the combination setting. So when we combine several drugs, specifically baclofen and methadone. So, and in this case, the risk of toxicity at baclofen in the left-hand side is a function of the dose of baclofen and dose of methadone that this patient receives that clinically prescribed by his treating clinician and parameter. So this is the approach that we have taken with the forwards trial. So one more advantage of using a model-based approach is that we could use an intuitively clear safety constraints and escalation decisions. So specifically from that model that we estimated that you've seen on the previous slide, we have extracted two main summary statistics for each dose of baclofen. Is that whether this combination of methadone and baclofen is safe, and this is defined as intuitive rule that if the probability that the risk of toxicity at the given combination of methadone and baclofen exceeds what is thought to be is upper toxicity bound 25%. So if the toxicity risk is above 25%, clinicians thought that it's too risky to administer this combination to patients. And this probability should be low for the combination to be unsafe. And if the likelihood that the risk exceeds its upper toxicity bound is greater than 25%, then we're saying that this combination is unsafe. And this concept was quite easy to communicate to clinicians that we deal with how likely the dose is safe or unsafe. And then we assign the next patient to that dose of next patient entering the trial, that dose of baclofen that is predicted to be the most beneficial for them. So, and the most beneficial mean that associated with the target risk of toxicity. So it was assumed that the sum risk of toxicity will be acceptable in this population, and clinicians decided that this is between 15 and 25%. So that combination that is most likely to have this risk of toxicity was prescribed to the next patient with this dose of methadone and baclofen. So it's currently ongoing trials. So we're currently recruiting, I think, our 4th cohort. So we're actively using this model to enhance our learning about whether it's safe to give baclofen and methadone together. And again, while there were some practical challenges and things to go through, this is one of the examples how we went through them and managed to implement and actively benefit from this model-based approach. Right. My second example will be linked to the question whether treatment works. So I think it was question 3 that Thomas mentioned today. An example here is the phase 2 group sequential trial called REBOR trial. So just a little bit of a background why an adaptive design was an option and what the indication is. So REBOR stands for resedative endovascular balloon occlusion of the aorta. And this is a little illustration for you. So essentially, a small balloon is inserted in the blood vessel to reduce the internal bleeding. So this is what it's supposed to do. So this is a device, and device is inserted in the above the injury. So one of the problems with the REBOR treatment is that there are only few observational studies available on it and no randomized controlled trials. But on the top of that, the observational studies conducted, they lead to quite controversial evidence. So a study conducted in Japan in 2015 found that REBOR is most likely to be harmful for patients. While another 2 studies in the US conducted in 2013 and 16 found that the REBOR is most likely to be beneficial, while another trial said that REBOR has no effect on the patients. So essentially, better evidence is needed to say whether REBOR is helpful for patients, or at least to some patients. So the trial to be conducted is a two-arm trial. So exactly the setting, we had a look before in the introduction section. So we have rebore and standard of care versus standard of care alone with no rebore intervention. So the primary endpoint was 90-day survival, and the current survival rate with standard of care only is 66.5%, and clinicians agreed that even slight improvements in this rate is interesting. But the problem is with the challenge with that trial was that the number of eligible patients per year in the whole UK is fewer than 80. And if we would like to do a conventional trial design, the frequencies and everything, we would need around 400 patients. So that would take quite a while. And when we're saying 80 eligible patients, we're just saying 80 in the whole country. It's not that who are ready to take part in the trial. So that would take quite a while to recruit 400 patients. So therefore, there were two requirements that this trial should have satisfied. So in order to become feasible, it should allow early stopping for futility. So we might, because there is controversial evidence, we would like to include an opportunity to stop the trial earlier and as a result, save some patients. And we also would like to benefit from the Bayesian analysis, given the stretch of the numbers we're facing in this setting. So the design that we thought is a good option, given the research question and the challenges, is the group sequential design that Thomas has spoken earlier today about. And this is an illustration how a group sequential trial could look like. So on the x-axis, we see the number of patients, and this example with 5 interim analyses. And at each interim analysis, we have futility criterion illustrated by a red bar. We have efficacy criterion that illustrated by green bar. And if we cross the green bar, we're saying that the treatment is efficacious and we can stop the trial earlier and conclude that this is superior to the standard of care. If we cross the red bar, we have crossed the futility bound, and we can already say that the treatment is no better than the control and we can stop the trial earlier. But if our performance is in the window, then we're saying that we don't have yet enough evidence to stop the trial, and we recruit next cohort of patients. And this is where we recruit N2 patients, again, standard of care, gray, control, standard of care or control in gray, experimental, reward treatment in red, and we compute our performance metric again. So the performance metric Z, this is Z statistics, which is based on the endpoint that we're using, that we're not getting in the technical details too much, but it can be quite general here. So one example of how a group sequential trial could have progressed is that we do our interim analysis at after first cohort of patient, we compute that statistic and we find that we are in the window, and we're saying with not enough evidence, we need to recruit more. We recruit more patients, compute that statistic again, and we again see that we're in the window, we're getting closer to the futility bound, but not quite yet. And we recruit more patients. And finally, after third cohort, we see that we got enough evidence to say that the treatment is no better than control, and we stop the trial earlier. And the obvious benefit of that is that we don't need to recruit these 2 more cohorts of patients, and we could save them from the treatment that has just proven to be no better than the control. So that would be unethical to continue the trial further. So applying this group sequential design to the rebore setting, that was a randomized 2-arm trial with 3 stages, so 3 interim analyses. So maximum number of patients was 120 patients, and interim analyses were at 40 and 80 patients, so equally spaced. And the decision-making was based again on the Bayesian criteria, the intuitively clear Bayesian criteria. So in terms of the log odds ratio of 90-day survival. So at the stages 1, 2, and 3, if we see that the probability that the treatment effect is negative, so that treatment, the experimental treatment is no better than control, given the data that we observed so far, and if this probability is high, 90% or more, we can conclude earlier that the treatment is does not work. So we have an opportunity to stop the trial earlier. We don't stop trial earlier for the efficacy reasons, only if we see that the trial, experimental treatment does not work. And for the final stage, we conclude that the experimental drug, the reward treatment, is superior to the standard of care alone if the probability that the treatment effect is positive, so the treatment does some good, given the data, is high enough. In this sense, is more, in this case, more than 95%. Again, this is currently ongoing trial. I think they are getting close to the second interim analysis, and the first interim analysis was conducted, and it was the recruitment was continued at that time. So the final example will be concerning the questions of what is the best treatment. And the example is the phase 2 multi-arm, multi-stage Taylor trial. So the Taylor trial is a study evaluating treatment for side effects of HIV. It was a superiority trial, and the objective was to establish which of the doses or dose of the treatment can offer benefits to the patients. And there was a continuous endpoint in this trial. Because there were several doses that were to be compared to the single standard of care, what we thought is the suitable trial design is the multi-arm, multi-stage design. And here is a quick illustration of what MUMS design can look like. So in the first picture, we have our first stage of the trial. We have gray patients here, the patients recruited to the control. And we have, in this example, 4 treatment groups. This could be different treatment, that could be different doses, which was the case for the Taylor study. So you can see the equally sized groups of 4 experimental treatments. So, and we have several interim analyses planned too in this example. So this is how we plan the trial. So this is the longest the trial could go. So we recruit the first cohort of patients to all our 5 groups for experimental and control and conduct interim analysis. In the interim analysis, imagine in this example, we learned that 2 cohorts of patients, for 2 treatments, the treatment were found no better than the control, and we could terminate recruitment to those arms in the future trial. So what you see here, we're not going to recruit to green and yellow treatment anymore because we found that it brings no benefits to patients. But for blue and red treatment, we have found no convincing evidence yet to stop them either for efficacy and futility, and we continue recruitment to these treatments. And then after recruiting cohort 2 and observing the outcomes, we conduct interim analysis, and we found that the treatment, the red treatment, is the good one and does provide benefit and superior to the control. So we can stop the trial earlier and recommend red treatment for further evaluation. So you see, we conducted interim analysis already. We found the superior treatment, and we don't need to recruit any further patients. So we stopped the trial earlier and saved some sample size in this case. So in terms of the actual Taylor trial, there were 3 active arms compared to the common control. There were equal randomization, so there was equal size of the groups, and there was 1 interim analysis to be planned in this trial. So opportunities at this interim analysis were to stop the trial earlier for superiority, exactly as in the previous example. If we found one treatment that is better, we recommend it for further evaluation, stop the current trial. We stop the trial if all treatment arms, all doses appear to be unpromising, so none of them seem to work. And finally, we can drop some of the active arms, not all, but only some, if they're not showing sufficiently promising signs of activity. And also an interim analysis as an opportunity to have a look at other trial and recruitment characteristics, for example, the dropout and recruitment rate, and they give opportunity to adjust it timely and appropriately. So the plan with this trial was that the decision what to do at the interim analysis will be made by the independent data monitoring committee that would follow pre-specified rules, and these rules were provided by the statisticians that did all the planning work, and the trial management group will accept these recommendations. So what happened in reality were that by the results of the IDMC meeting, 2 arms were recommended to be stopped. So that was the recommendation to Team G. But Team G wanted to see unblighted data before confirming. At that time, that led to very lengthy discussions about what this blinding would give and what alternative decisions could be. And essentially, the argument was that if 2 treatments had already crossed the futility bound at the interim analysis, the chances that they will be found to be good at the final analysis are so low that there is no ethical basis to continue with 2 treatments. So this decision was taken forward. And one lesson that we learned from that implementation is that the Team G should understand the decision process crystal clear and should be happy and buy into the stopping rules and the stopping criteria. So all of that should be discussed, of course, in advance, and everybody should sign up for that. Right. And the final example will be a platform trial example, for which I will hand over back to Thomas, because this is the trial that he has been involved the most. So Thomas, back to you.
Thanks. Do you just want to stay on the slides and advance?
Yeah, will do.
So I'll be talking about the recovery trial, which is a multi-arm platform trial for COVID treatments. Initially, the trial started off with 4 active treatments compared to standard of care and had mortality at 28 days as the primary endpoint. One of the sort of innovative features of the trial was that it minimized data collection and used data linkage to electronic health records to obtain many of the relevant data for the study. Next slide. It's been a great story, and you might have heard of it, in that it recruited over 11,000 patients in the first 3 months. It enrolled 175 centers in that same time. It's a trial that's open to all ages, and it was a trial that went from funding to recruiting first patients in 9 days. So let's have a look how that trial looked like. Next slide. So this is what the recovery study looked like on the 19th of March 2020. So you can see the 4 different experimental treatments: hydroxychloroquine, dexamethasone, lopinavir, ritonavir, and azithromycin, all compared against the standard of care at the top. Next. About a month later, an extra arm was added, tocilizumab, to the comparison. So you can see immediately here the benefit of the platform structure, in that as new evidence emerged, we had the opportunity to add new treatments to the trial. So we could use the same infrastructure that was already in place, the same 175 centers that were in place in the first place, to recruit additional patients to that one comparison, to that extra comparison, I should say. Next slide. Another month later, roughly, a second arm was added, convalescent plasma. Next slide. And about a week later, we had the first decision. So again, you can see the efficiency of that platform structure, in that it was found that hydroxychloroquine was not yielding benefit in that particular patient group, and hence the trial, that comparison was stopped for lack of benefit. Next slide. Another 10 days passed and we got our first winner. Dexamethasone was shown to improve survival of COVID patients. And again, you can see, because of the platform structure, we could make decisions as quickly as it emerged, and therefore made sure that the treatments were available to patients as quickly as possible, made available to patients as quickly as possible. Next slide. And you can see the trial went on, and I'm not going to talk you through the whole story of the study, but at the end of the day, up until this week, there have been 10 different treatments investigated through that single platform trial. 4 of them were shown to have benefit to patients, at least in some group of patients, while we also concluded that 6 of the treatment that, based on preliminary evidence, were deemed to be potentially helpful, did not show sufficient benefit to warrant rollout on a broader basis. And with that, I think, Paul, can you just check? Yeah, there's one more treatment that we've stopped here on the slide. But I think with that, we'll stop us talking and give you the opportunity to ask us any questions that you might have.
Okay, thanks a lot, Thomas and Paul. Really had a very nice talk. I'm sure the audience has got important information that might make their trial quite efficient. Now the floor is open for Q&A, and if there are questions, Q&A, looks there are no questions so far, then you could ask verbally as well.
Yeah, while people think about their questions, I just realized, talking through these, or listening through the examples again, that none of the examples that we've spoken about were in a resource constrained setting, if you want. And just to reassure and to mention that there are these types of designs have been used and are being used and are increasingly being used also in that context. A couple of tuberculosis trials, for example, that have used a multi-arm, multi-stage design. We're currently in the process of starting an HIV study in sub-Saharan Africa that's using a platform approach. So even though the examples that we've shown were all all of them based out of the UK, there's nothing specific about them being run in the UK that can be run, and there are successful examples of these studies elsewhere as well.
Thank you, Thomas. Okay, right. So, will there be any question? Now the floor is open for discussion.
Professor Yasu, while the participants in the audience prepare their questions,
yes, please go ahead.
Some questions, and if I can ask.
Please.
Yeah, so thank you very much, Professor Thomas and Dr. Pavel, for the nice and interesting presentations. Professor Thomas, you have already preempted one of my questions, whether you know what the experience looks like in Africa. And the other question that I have is that definitely we expect some challenge in the African setup or in the developing setups. So from your experience, I know you are working more on the design aspect and not in the implementations and operations, but just in case if you are aware, what could be the potential, particular challenge in African setup or developing country setup? And probably also if you can add how we can also tackle these problems. Thank you.
Thank you, David. Maybe Thomas, you may answer them together. If I may add one more question. Yes. Is that okay? Okay. Okay.
So,
may I add? May I ask as well, so that you could respond both together? Okay. Thomas, would it be ethical to increase the sample size beyond the calculated one? Is that considered to be ethical?
Okay, I'll start with the second question. If you have a study that is adequately powered, increasing the sample size is probably not ethical, because you should have sufficient information to make a decision on the basis of the data that you have at hand, and therefore increasing the sample size would imply that you effectively withholding treatment for patients outside of the study, or potentially withhold the treatment for patients outside of the study. That, to some extent, is probably the lesser problem. The bigger problem is, unfortunately, many treatments don't work. And so by increasing the study, even though you could conclude already whether or not the treatment is successful, you potentially exposing additional patients to a treatment that isn't working. And as I said, unfortunately, more treatments don't work than do work. And so I would not consider it ethical for an adequately powered study to increase the sample size, while at the same time, I don't think it's ethical to run an underpowered study, and hence my call to at least consider whether sample size reassessment or reestimation can be used in your studies to make sure that at the end of the day we get a valid and definitive conclusion. Pavel, anything to add from your end?
No, nothing particularly.
Now it's question then.
So then, with respect to particular challenges in the African setting, so I mean one potential challenge, which is probably not necessarily unique to Africa, but might appear more often, is obviously regulators and ethics boards are going to have to look at the design. And if this is a very traditional two-arm randomized trial that these bodies have seen numerous times before, it will be much, much easier for them to make a decision about the suitability of the study. While if you use an innovative design that they are less familiar with, you might expect not necessarily pushback, but more need to clarify what you're intending to do. So in our experience, it might mean that you have an extra meeting, an extra discussion, an extra response that's required in order to just clarify why you think this is the right thing to do and what the benefits and drawbacks are. The other thing that I found to be possibly more problematic in some African settings is ensuring that patients are able to take part in the study. What I mean by that is that in rural areas, the patients will have to come to whatever center it is that runs the study, and therefore it becomes more difficult for them to take that journey on them, to get to the center, to take part. And in some cases, you're excluding whole groups of patients from even considering participating in the study because they simply can't get to the centers where the studies are run. So again, as you've mentioned before, I mean, this is not my main area of expertise, but it's something that I've seen a couple of times now, that more emphasis is placed on making sure that patients get the support to stay in the study and travel to the places that they need to travel. And that might mean that travel is organized, that travel vouchers are being made available, that childcare is provided for the half day that it takes to take part, and so on and so forth. So there are different things that one can consider that unfortunately are all adding to costs of the study, but it's definitely something that I've learned is more difficult in some African settings.
If I might add.
Yes, Pavel, you can add, yeah.
Yeah, so I guess one more thing that I can think about, and again, it might not be very only specific to Africa, but it might put an additional stretch on already stretched resources, is the actual implementation, because we thought about, designing and how you do all the preparation work, but somebody have to do that work and put time and resources into that. And while we're talking innovative designs, many of the options that we talked about today, they're well studied. And what is important, they are already implemented in software. And so that obviously can help with implementation and with a stretch of the resources. And there are tools like, the shiny apps, so, web-based tools where you don't need to look at the code, but where you can press buttons and the nice interactive software that one can play with and use to design a trial. So hopefully there is some work is being done. So for example, we're aware of one dose-finding trial, so that used innovative model-based design similar to what we talked about today in an African setting, using software that was developed by our group, interactive software. So there are definitely examples of how this could help with implementation.
Thank you, Thomas and Pavel. There are a few questions on Q&A. I might need to read. Thank you so much for the wonderful presentation, Prof. Thomas. You said there are chances to conduct platform trials in SSA. Are there any consideration in the ethnic and genetic composition in these different settings from the UK? That's a question raised by Charity. Shall I read them all, or shall you respond one by one?
Yeah, probably easier to respond one by one.
Okay. Okay.
It's a very good question. I think the problem or challenge of diversity, if you want, is there in any trial. Now, clearly, the UK population is going to be more homogeneous than the population in a whole continent, and so therefore, it is essentially, in my view, to at least explore whether or not there are sort of other aspects. Could be genetic composition. It could be environment that cause these differences. In my view, the problem is, however, not very different from multinational trials in the first place. Quite a lot of studies are run across multiple countries, and there as well, I think one of the standard analyses that one does is to make sure that there are no meaningful, or not, try to understand whether there are meaningful country differences, and I would expect the same to be necessary in a platform study across well, any multiple country setup or multiple region setup.
Thank you, Thomas. Would you like to add, Pavel, or? Shall I move on to the next question?
No, that's good.
Okay, thank you then. Okay. The next question reads, please, why was your endpoint mortality for the fourth study rather than reduced fever, viral load, etc.? So the question raised by Adela.
It's a good question. I mean, it's twofold. The one argument is that changes in viral load, for example, are only meaningful if they also yield improvement on a clinically meaningful outcome. And in that case, the clinically meaningful outcome was deemed to be mortality. So that's the one reason. The other, which in my view is probably the more relevant in this context, the study started in March 2020, so only about 3 months into the first, after the first cases of COVID had been detected. So we just didn't know enough about viral loads and burden for fever and how many patients didn't get fever or didn't get high enough fever and so on and so forth, that we just didn't trust these kind of metrics. Now, 2 years in, we have a much better understanding, and we understand much better what a meaningful change in viral load would be, and hence that might be a useful endpoint to consider, and is in fact considered for some of the phase 2 evaluations now.
And if I may add, and please, Thomas, do correct me if I'm wrong here, I guess it was also about resources at the time, because that would be conducted in the same NHS hospitals that were overloaded at the beginning of the pandemic. So you don't want to overstretch your staff with additional measurements, additional tests. And I mean, it's cynical that, as it may, sound, that the mortality is something that would be measured anyway, that would not put additional stretch on the resources. I think that was also a consideration at the time.
Absolutely, yeah. Thanks, Pavel.
Thank you, Pavel and Thomas. The next question is raised by Shifarao. When exactly and how many interim analyses can be done in adaptive design? Any of you who respond?
Pavel, do you want to have a go?
I mean, to be honest, as many as you wish. And but it's always a trade-off on what is meaningful. And as we add more interim analysis, we